Hello my dear students, welcome to the MS Science Academy. Myself Minu Srivastava, and today we are going to discuss about the transport across the cell membrane. As I have not launched so many videos from so many times, so for that I'm sorry. But now we are going to start the human anatomy and physiology chapter subject, and in that nursing, and we will cover the nursing and the pharmacy and the BSc as well. In that the anatomical positions and the introductional lectures like cell, that lectures I have taken, okay, and I have uploaded previously. So from that I will continue, and today we are going to discuss about the transport mechanism. The transport means the substance transport via the transport membrane or the via across the transport across the membrane. Okay, so I will write the definition. Substance move across the cell membrane via transport process, via various transport process. Okay, so in this process two main process, one is active and second is the passive process. So here two processes, passive process and active process, means the process the transport mechanism involve the two type of the process. One is the passive process and second is the active process. In the passive process, as the name suggests, it is the passive, means it has no requirement of energy, means it will occur without the utilization of energy, no need of energy, whereas in the active process the energy is required. And you know that the cell's energy currency, that is ATP. Okay, so no need of ATP, but they need the ATP. The full form of ATP is the adenosine triphosphate. Okay, so here the passive process, as the passive process didn't require others energy, the particles move according to their own kinetic energy. Okay, so here the passive process is moved according to the concentration gradient, according to the concentration gradient. Gradient means the difference. The gradient means difference, means here the particles are so many, like 100 particles, and here the particles are 2 or 3 particles. So the difference is known as the concentration gradient or concentration difference. Okay, so here in the passive process, the movement of the particles according to the concentration gradient. Okay, so this process is known as the downhill process, because the as you know, when you are just pouring the water from top to bottom, there is no requirement of motor or the electricity energy. So this automatically the water moves from the upper side to the lower side. So this is the downhill process, and here no concentration gradient required. Means if there is a less particles also, with the help of ATP, with the help of the other energy, we will move the particle according to their less concentration also. Okay, we will see in that active process when we will discuss later on. Okay, so here that it is a downhill process where it is known as the uphill process, because as we want to move and we want to move the water from lower side to the upper side, we have to use the motor energy or the electricity energy. So that process is known as the uphill process, because we want to move the particles from the upper side, okay, from the higher side. So that is known that they will require the energy, so it is known as the uphill process. Okay, here the downhill process and the uphill process. Now the examples is diffusion, osmosis, facilitate diffusion. Okay, and whereas the active transports example is the primary active transport, secondary active transport, primary active transport process, secondary active transport process, exocytosis and endocytosis. Exocytosis and endocytosis. Now one by one we will discuss about these 5 topics. These 1, 2, 3, 4, 5, 6, 7. 7 topics we will discuss one by one. So now let's start with the diffusion. As the name suggests, the diffusion, the diffuse out means that something will diffuse out. Means that something or the particles will diffuse from the higher concentration to the lower concentration. Means that according to the concentration gradient. So now come to the diffusion part. The diffusion is the passive process. As we know, the diffusion is the passive process, as we have discussed just now. It's a passive process and the particles move according to the concentration, gradient. Okay, the particles move according to the concentration gradient. Okay, and here the concentration gradient means the material. Suppose that here the particles are 100 particles and the other side the particles has 2 or 3 particles. So automatically the particles will move from the higher concentration, to the lower concentration. Okay, so we will write the definition that the movement of the particle from its high concentration to the low concentration. I will write the definition. Movement of particles from high concentration to the low concentration, Okay, so here the particles move from the low concentration to the high concentration. Okay, so here the some factors responsible for the movement of the particles. So there are here is the factors. First factor is that temperature. If the best example of the diffusion is that if your mama cook kheer, okay, that kheer or pudding in the kitchen, so that kheer or the pudding aroma, okay, smell will reach to the drawing room, to the dining hall, to the bedroom, everywhere, because the particles are in high concentration in the kitchen. Okay, so particles automatically move from the high concentration to the low concentration. Here one more thing is that the particles move with their own kinetic energy. So don't be confused with that. Okay, here once you told that it's a passive process, so the passive process means they will not need the others' energy. They will have their own energy and according to their energy they will move. Okay, so particles move with their own energy. Own kinetic energy. Okay, so movement of the particle from the higher concentration to the lower concentration according to their with the use of their own kinetic energy. So here the first factor is the temperature. So if the temperature is high, the automatically rate of diffusion is high. Means the aroma of kheer will reach very much faster in the summer period as compared to winter. Okay, so the temperature, if the temperature is high, the rate of diffusion will be more. ROD means the rate of diffusion. Second, the steepness of the concentration. Steepness means sharpness or the difference. Means I will explain the steepness of concentration gradient. Here, if the particles is 100 and here on the second side the particles is 2 or 3, so the concentration difference is more and more higher. Suppose that if the particle one side is 100 and the other side is 80, so the difference is not higher, so the movement of the particle is slower. Okay, so if the concentration gradient is more steep or the more sharp, then the rate of diffusion will be higher. So here the steepness is the high. If the steepness or the difference of the concentration gradient will be higher, then the rate of diffusion will be higher. Okay. Now the next one is that the steepness, first is the temperature, second is the steepness, and third one is the mass. Okay, if the particle mass of particles, if the particles having the more mass like me, so the rate of diffusion will be slower because the particles will not move fastly. Okay, so here the mass of the if the mass of the particle will be higher, then the rate of diffusion will be higher. Okay, so and if the if the mass of the particle will be less, okay, so the rate of diffusion will be higher. It means the particles if are with the less weight or the less weighter, then the rate of diffusion will be higher. Okay, and the last one, that is the surface area. If the surface area is higher, means the particles get more space to move fastly, they can run fastly. Okay, if they not get the space, okay, so that will constricted path, they will if they get the constricted path, then the rate of diffusion will be lower. So if the surface area higher, then the rate of diffusion will be higher. Okay, now this is the diffusion part. Now the diffusion will be with the according to the membrane, we will see the diffusion with the help of two subtypes: diffusion through the lipid bilayer and diffusion of the particles through the ion channels. Means what I want to say, then in our body system or in the cell, the particles of the diffusion of the particles according, or the particles of the movement by two ways. Okay, first is through the lipid bilayer. Okay, for that polar hydrophobic molecules diffused freely through the lipid bilayer. Okay, and second, some particles move through the ion channels. So first is the movement through the what is the topic is that movement of the particles through the membrane, diffusion through the membrane. First is through the lipid bilayer and second through the ion channels. Okay, so here in the polar hydrophobic molecules, here the polar hydrophobic molecules move molecules diffuse freely through the lipid bilayer, diffuse freely through the lipid bilayer, LBL, lipid bilayer. Okay, lipid bilayer. Okay, and without the help of the membrane, if the molecule is the polar and hydrophobic in nature, they do not use or do not want any kind of the help of the membrane transport protein. Without help of membrane transport protein. Without the help of membrane transport protein. Okay, so here first movement, polar hydrophobic molecule easily move through the lipid bilayer, and if the substances are not polar hydrophobic in nature, they are the other kind of the nature, they will move through the ion channels. Okay, other than the hydrophobic molecules move through ion channels like potassium channels, sodium channels, calcium channels, like that. Okay, chloride channels, like that the different channels will move. Okay, so now we will come to the facilitated diffusion. In the facilitated diffusion, we will see that the carrier proteins or the other carrier molecules help to diffuse out the material from the one side of the membrane to the second side of the membrane. Now come to the facilitated diffusion. As the name suggests, some particular molecules facilitate such kind of the diffusion, and these facilitated molecules or the molecules which facilitate such kind of the diffusion, these are some carrier molecules or the carrier proteins. Okay, so the diffusion through the specialized carrier molecules bind with the substance like lock and key. Okay, so we'll write the definition. Diffusion through specialized, okay, diffusion through specialized carrier molecules, specialized carrier molecule which bind with the substance which bind with the substance like lock and key. Lock and key. They change their shape and deposit the material and deposit the substance to either side of membrane. Okay, so here the particular carrier molecules are using in such kind of the diffusion, and these carrier molecules bind with the particular substance which is to be diffused out and diffused in or diffused out. Okay, so here the carrier changes the shape and deposits the substance on the either side of the membrane. So here we will see, if suppose that this is the lipid bilayer, this is the lipid bilayer, this is lipid bilayer. Okay, so here the carrier proteins or the carrier molecules bind with particular substances, bind with the particular substance by changing their shape. Means some changes will occur in their shape so that the molecule will bind in that carrier molecules and they will diffuse out to the either side of the membrane. Okay, they will diffuse out to the either side of the membrane, either the diffuse in or diffuse out. So this kind of the diffusion is known as the facilitated diffusion. Now come to the osmosis. Now come to the osmosis part. Osmosis means the movement of the solvent from its high concentration to the low concentration through the semi-permeable membrane. Okay, so here the main thing is that the particles will move, solvent particles will move. Who will move? The solvent particles will move from the high concentration to the low concentration, but through the semi-permeable word you must be used. If you are not using this line through the semi-permeable membrane, if you are not using this point in your definition, then the automatically examiner will cut your marks, because if you are not writing this, only you will write the movement of the solvent from its high concentration to low concentration, so automatically it will similar with the diffusion. Okay, so here through the semi-permeable membrane you have to justify that the osmosis through the semi-permeable membrane is the very much essential one. Okay, so here the osmosis is the movement of the solvent particle from its high concentration to the low concentration, okay, through the semi-permeable membrane. So according to the concentration, there are three types of the particles or three types of the solution. First is the isotonic solution, second hypotonic solution, and third one, that is hypertonic solution. The first thing is that isotonic, hypotonic and the hypertonic, these three terms are the comparatively terms. isotonic, hypotonic and the hypertonic. If we are saying the solution is isotonic, so that we have to discuss or describe that in which reference it is the isotonic. If we are saying that it is the hypo or hypertonic, so we have to mention that in which reference it is the hyper or hypotonic. So that will be our main consideration, that isotonic, hypertonic and the hypotonic terms are the basic comparative terms. So we have to compare the two solution. If we are comparing the two solution, here suppose that solute is 2 gram and the solvent is 100 ml, and these are differentiated or separated through SPM, semi-permeable membrane, and in the second chamber the same concentration 2 gram and the 100 ml we will keeping. Okay, so these both chambers are basically isotonic to each other. These means these both chamber are isotonic to each other, so there will be no movement of the solvent will be shown. Okay, now second one is the hypotonic. Here if we are separating the two chambers through the semi-permeable membrane, and if we are taking the solute 2 gram and the solvent is 100 ml, where if we are taking here the solute is 4 gram and the solvent is lesser, like 80 ml, like that. So here the first A solution and the B solution, if we are comparing the A and the B solution, the A solution is the hypotonic solution. The A solution is the hypotonic solution because here the solvent concentration is higher, so the A solution is hypotonic solution, whereas the B solution is hypertonic solution because the solvent concentration is lesser. Whereas in the hypotonic solution the solvent concentration is higher. So here in the hypotonic solution, the movement of the particles, of the solvent particles from the hypotonic solution to the hypertonic solution, because in the definition as we have discussed, the movement of the solvent from its high concentration, the concentration is high in the A chamber and the particles of the solvent will move from A side to the B side through the semi-permeable membrane. So this is the, if we are taking the hypotonic solution, the movement of the solvent from the high concentration means from hypotonic solution to the hypertonic solution. Now we will see with the example of one RBC example, then what will happen if we are taking the RBCs in the isotonic solution. Suppose that if we are taking the RBCs in the isotonic solution, then there is no change. Because as we have discussed that in the isotonic solution there is a no movement, so the RBC shape will not change. Now in the second one, if we are taking the RBCs in the hypotonic solution, means the water concentration is more in the test tube as compared to RBCs. If we are taking the RBCs in the hypotonic solution, means what it means in the hypotonic solution means the RBCs water concentration, the water molecule which present inside the RBCs is lesser as compared to the water molecule present outside the RBCs or the surrounding the RBCs. So this solution is hypotonic solution. So in the hypotonic solution, so movement of the water molecule from its high concentration to the low concentration, the movement of the water molecule from its high concentration to the low concentration, and the RBCs will swell up. Okay, so the RBCs will swell up. Now what will happen if we are taking the RBCs in the hypertonic solution? Hypertonic solution means the water molecule surrounding the RBCs is lesser as compared to the water molecule inside the RBCs. Okay, so automatically what will happen? The molecule of the water will move out and the shrinkage of the RBCs, means the RBCs get shrinked out. Okay, so like that, the movement of the water molecules occur through the semi-permeable membrane, and these hold for the osmosis. Just you have to remember the one thing, that the 0.9% NaCl solution is the normal saline solution, which is the isotonic for RBCs. What I am saying, that the 0.9% NaCl solution is the isotonic for the RBCs. And what is the isotonicity, and how you will prepare the 0.9% solution? For this, you have to take the 0.9 gram RBCs, 0.9 gram salt, NaCl, and 100 ml water. 100 ml water means in the 100 ml water you will put, or you will dissolve the 0.9 gram NaCl, so that 0.9% NaCl solution will be prepared, and it is the isotonic for the RBCs. So here this is all for the passive transport. Now we will discuss about the active transport.